Well, we're back. We're back with session 5, clinical translation, registration, registrable indications. And we're here to talk about regulatory endpoints that are actually now being pursued or can be. We've got 3 terrific panelists who are providing different perspectives, but the great thing about them is that they, in their own way, are actually in the game. They are pursuing indications in professional programs that can ultimately serve as metabesity approaches. So first, let me introduce my long-term friend, Joe Cook, who, as CEO of Amylin, introduced to the world of diabetes a game-changing class of drugs, the GLP-1 agonist, which in themselves could be considered metabesity solutions. So Joe brings that great contribution, but he has not rested there. He's currently CEO of NuSirt, another company that is pursuing metabesity approaches. Joe, I believe you have some slides, and then I'll introduce Joan and James.
Yes. All right. Can you hear me okay?
Hear you great.
Okay. If you'd put up the first slide, then we can start. While that's coming up, NuSirt is a biopharmaceutical company that was formed on the basis of some work that Michael Zimmel had done in his 30 years of studying energy, and concluded that it was important to try to figure out a way to enhance the sensitivity of the SIRT1 receptor system, and was focused on how to accomplish that, and started out with a variety of ideas, but narrowed them down as he examined the impact. I don't see, are the slides up? I can't see them on my screen. Looks like they're starting. Okay. So next slide, please. So the vision and goals of NuSirt really focused on, can we prevent or treat age-related diseases using already characterized compounds with established safety profiles by focusing on this energy sensing system, the SIRT1 pathway. We were intrigued with the notion that early preclinical work pointed to the fact that we could shift the NAD curve, the sensitivity of the curve to the left, meaning that enhanced values or enhanced benefit from NAD, we could enhance the benefits with a given quantity of NAD plus by shifting the sensitivity curve, by amplifying the signals in the SIRT1 pathway. Michael's the leading thought processor on this and has really worked hard. We then developed an approach to try to understand, could we use this observation that was confirmed in augmenting the utility of pharmaceutical combinations with leucine in obesities. And by the way, leucine was found to be the only branch chain amino acid that actually enhanced this SIRT1 pathway sensitivity. And our first target was to try to see if we could develop a more user-friendly version of metformin by adding leucine to metformin and seeing if we could achieve the same level of glucose control with a lower dose of metformin plus leucine compared to the labeled doses of diabetes. And that result in type 2 diabetes subjects showed that there was a non-inferiority performance, meaning that adding leucine to a low level metformin produced essentially the same results as the label claim dose, which was higher. Then from that, however, we decided that there was some important signals, and then we conducted a couple of studies, or one study actually, in non-alcoholic fatty liver disease, where we studied individuals and found that by adding leucine and metformin, plus a third component, sildenafil, we are able to focus on improving liver fat. So next slide, please. I'll get to some of the results in a minute. This began to shape in our mind a view of this whole constellation of impaired energy sensing, largely around AMPK and SIRT1. And it, as everybody I'm sure on this panel knows, it seems to be pervasive in its impact. And as we've talked before, number one, if you can simply lower weight, you have a tremendously improved energy sensing system. But we were trying to find if there is a way to modify this by adding leucine to co-activating compounds. Next slide, please. Because regardless of how we've tried and how many messages have been sent, weight loss is not something that simply most people get onto. So our idea was to add leucine, which amplifies the sensitivity of SIRT1 to co-activating compounds. And in this model, we determined that we need not only to have an AMPK agent such as metformin, but also we needed to increase eNOS. And that led to the next slide, the idea that we can enhance this work by coming up with a three-part solution. One, leucine that activates SIRT1, metformin that activates AMPK, and a very small dose of sildenafil. Notice that's 1 milligram. All these doses are BID, which activates eNOS. It's interesting, sildenafil is the only one of the PDE5 inhibitors that does this, and we're not interested in the PDE5 inhibition because at 1 milligram BID, you get very little of that, if any. We were more interested in the activation of eNOS. Combination of those three agents we then studied in NAFLD patients and also in obese patients. So go to the next slide. So in 2 clinical trials, we've conducted 3 clinical trials, one in diabetes with a dual combination of leucine and metformin, and one in NAFLD, non-alcoholic fatty liver disease patients, and one in obese patients. So what you have here is a composite of the results that we have shown. One, a fairly marked and very dose-related response in weight loss. That's in the upper left panel. Placebo-adjusted weight change over a 6-month study, or 24-week study, showing the various doses, which led us to the target dose of leucine plus metformin plus sildenafil 1.0. We did not see any material improvement in adding more sildenafil. In fact, one might argue a slight trend upward. We then said, well, what's going on with subjects who have higher levels of insulin? Back to the comments that were made earlier about insulin resistance. While insulin levels greater than 12, we saw a more profound change in weight. That was encouraging to us because it seemed to be compatible with our profile of how we were working. Then in the NAFLD study, we looked to see for those subjects that presented with abnormal ALT, that is greater than 50, did we see a change in the percent of fat as measured by MRI PDFF? And we did indeed see that and I'm sorry the legend dropped off, but the C refers to leucine sildenafil metformin combination. Other observations we made was a 5.5 drop in mercury in diastolic overall, and quite frankly, a higher drop in hypertensive and an improvement in triglycerides and a reduction in HOMA. So our next slide. Our combination of those results we were excited about, but as we tried to find a pharmaceutical level partner, we wound up with a lot of comments back. And by the way, we have all of these very well covered with global patents. We spent a lot of time to cover the mechanistic as well as composition of matter patents. Unfortunately, from a large pharma standpoint, many said, well, I only want new molecular entities. You're dealing with old drugs, metformin and sildenafil. Somebody will do a workaround. So our current thinking is to try to move to the left on the continuum of the solution spectrum. So I'll draw your eyes to the middle arrow. On the left is this multiple nutraceutical players with unsupported noise, and on the right is a highly regulated legend drug space. We are now focused on developing the knowledge that we have from these clinical trials into natural products, provided we can deliver what we believe are 4 must-haves: published evidence in the clinical space, a safety stamp that is without question, a rigorous method to determine that you have what you have in the product and it's not just that collection, and a presumption of an MOA that's established and well accepted. So that's my that's my overview of the journey we've been on. We have shared a lot of frustration with our data seemingly pointing in a positive way, but the appetite isn't quite there. So we're trying to find if there's an alternative way to work this through what I would call a quasi-regulated natural product space. Back to you, Zan.
All right, Joe, terrific. That was, as usual, superb. We'll come back to some of your lessons learned in a moment. But I would like to bring Joan Mannick in, who brings really a remarkable training and career. As it turns out, she was trained at Harvard as an infectious disease specialist. I didn't even know that. I thought she was sort of in my silo of endocrinology. But she has spent a good while in big pharma, and more recently as chief medical officer of ResTORbio, and is responsible for the first really what you could call Metabesity trial that has provided real clinical outcome data. So nobody is better than Joan Mannick to talk about her particular approach that she's taking in the COVID era to enhance the immune function of people who have age-related decline in immune function.
Thanks so much, Zan. So I'm talking in a later session about our trial for enhancing immune function for COVID-19, and I thought in this session I'd more just give a 50,000-foot overview about how we think about taking the great basic science that we've heard about in the talks earlier this morning and bringing them to the clinic. So next slide. So questions that I think most people in biotech and pharma think about when thinking about how to translate aging biology to the clinic are, one, what are the best validated mechanisms underlying aging biology? Two, are the mechanisms associated with something which we call apockable indication? And that is, is it possible to do a relatively short, inexpensive phase 2 proof of concept trial that will enable a company to make an early go, no-go decision to de-risk costly late-stage development? And some indications have these proof of concepts that you can do and others don't. So we want to veer to the indications where we can do these kind of de-risking proof of concepts. 3, of course, is there an unmet medical need? 4, which we're going to focus on in this session, and I think it'll be great to hear Peter Stein later, what is the regulatory path to approval? And then last, what a lot of people don't think about, but is really important when you're picking your registration endpoint, will payers pay for the drug with that particular endpoint and indication? So next slide. Just quickly, there are now well-validated mechanisms that have been established that underlie why we age. And what's exciting is each of these mechanisms now can be targeted therapeutically, and I think this has the potential to transform the practice of medicine, because as we've heard, this aging biology is a common denominator that is the greatest risk factor for most chronic diseases, and we've never approached these diseases by targeting this biology. So next slide. So once we've picked that indication or that mechanism, we have to go, if we target that mechanism, can we do a proof of concept trial to see if targeting that mechanism has benefits in a human? And the reason we need to do inexpensive proof of concept trials is the cost of developing prescription drugs through getting marketing approval is approximately $2.6 billion now. So companies cannot afford to get to late stage development and have frequent failures. It's just too expensive. So that's why we need to figure out proof of concept trials that de-risk the very expensive late stage trials and just kill quickly drugs that aren't translating to humans and aren't likely to work. So next slide. And then we have to figure out if we have an indication and it has a proof of concept pathway, what is going to be the indication for which we can get marketing approval from health authorities. And what's a simple way to think about this is that regulators need to be shown that your clinical benefits of a drug outweigh the risks. And I think what's important in the aging field is that the endpoints need to be, at least for the FDA, something that reflect how people feel and function. It's not enough to say, oh, we've moved an inflammatory marker, or, oh, you know, some lab test has gotten better. It has to be a clinical functional endpoint that's relative, that is related to how people are actually functioning. That's what's needed to get your drug approved. And I think another really important thing is that we are going to be moving into some indications for which there is no regulatory path that has been established. And those new regulatory paths, there's going to be a learning curve, and we're not always going to get it right in the first few trials. And I can just use resTORbio as an example. As Zan said, we're developing a drug that enhances pan-antiviral immunity in the elderly. So it's not specific for one virus. If we can enhance antiviral immunity in older people, it can decrease the incidence of all sorts of different viruses. The FDA has never had a drug like that where they have granted approval. So they had to work out what's the right endpoint for this. They changed their mind between the phase 2B and phase 3 trial as they tried to work this through. And as we started getting data, it was clear that there's probably going to be a more personalized approach that's going to be required for some of these anti-aging endpoints. And we're going to have to identify who really has aging-related deficits and who doesn't. And age greater than or equal to 65 is probably not specific enough to sort of identify the populations who are going to have the greatest benefit from drugs targeting aging pathways. And then last slide, next slide. Really important is to think, is this indication something that payers and medical insurers, particularly Medicare, is going to pay for? Because if Medicare does not pay for a therapy, then older adults are not going to have access to it. So payers are have strapped budgets. The over 65 population is the fastest growing population in the world, and it's an expensive population. So for payers to be willing to reimburse for the cost of a therapy, we really need to show there's compelling data that this is really of clinical benefit to a 65 and older population. It's not just a Me Too drug. And something people don't think about, it's important to show that whatever this cost of this drug is, it's offset by saving cost on things like hospitalizations. So I think I'll stop with that, Zan, and we can discuss more after.
Well, that was great, Joan. Thank you very much. We'll, of course, come back to a discussion in a moment, but let me bring in Dr. James Peyer, who brings a hard scientific background as an NSF fellow, and then later a McKinsey consultant, and more recently a serial entrepreneur and venture capitalist. He's working for innovative ways of pursuing metabesity therapies and using a stepping stone approach to get there. So James, thank you. Go right ahead.
Absolutely, Zen. And I don't have any slides to share today. So maybe a couple of comments and then we can launch into the group discussion. I think I take a slightly different approach to this whole field of geroscience than Joe and Joan have, because the group that I started both in my time as a venture capitalist and now as the head of a new company that's still partially in stealth mode called Cambrian Bio, we're really focusing on those NCEs that Joe was talking about, the new chemical entities targeting the different aspects of the biology of aging. And the issue, the challenge of this approach is that you start much earlier in the developmental phase. You can't jump straight into into clinical trials when you're starting work on brand new mechanisms and trying to identify completely new drugs. So we have, 5+ years from beginning a program until we're going to be starting our pockable, our phase 2A proof of concept trials. And my view is that because this topic of going after aging biology is so new, that there will be some opportunities for decent sized effects from repurposing of existing mechanisms, but really starting brand new programs, rethinking the way we're doing drug development and tearing apart the mechanisms with new entities is going to be the way that we see, really big effects. But in order to do that, we have to take a very long view, and we have to be ready to brace for many failures along the way. And so Cambrian's approach, as opposed to starting with just one drug or one mechanism, is to plant early stage seeds across that whole spectrum of the different types of damage that accumulate as we age, the 9 hallmarks. And so currently we have 9 programs under development, ranging from one target to hit program, but then a lot in the lead optimization stages up through IND enabling studies. And as we scale, we're going to be taking 20 or more shots on goals on lots of different mechanisms in order to find the first drugs that have this potential to be geroprotective, but can be NCEs that pharma will want to work with and have the patent lifespans to do what Zan alluded to, what I call a stepping stone indication approach, where our pockable trials can be almost anything, ranging from specific cancers in the oncology space, so like we have some specialty oncology drugs, to orphan diseases, or even going after big age-related disease indications, although that's the rare case for us. And then after those drugs show success in a phase 2A or even phase 2B larger trials, then we can pivot those mechanisms, devote additional resources to doing large trials with sort of that metabesity theme, which is how I think a lot of our portfolio is going to play out. So that is a bit vague, but some general comments about a different approach that I've taken to addressing this field.
So actually, James, I think that's perfect. It does contrast with, say, Joe Cook's approach, repurposing already approved drugs, although in a proprietary combination. To some extent, it contrasts with Joan's approach, which is going right after what could be considered age-related indication. So let's have some back and forth between you three. I haven't heard any violent disagreement in the validity of each approach. What do you have to say, Joe, about the approach that James is taking, which is going to be a longer game?
I'd say come one, come all. I mean, I don't think any of us are smart enough right now to figure out whether one's superior to the other. So let's all go at this thing. I think the problem is so big and it's not going to lend itself. I remember early in my life of working in drug development, an admonition by a very sage old person. He said, look, Joe, he said, you got to understand something as fundamental as energy. There are no single cause and effects. Because if such existed, they probably didn't survive to have children. So we are the progeny of people who have compensating mechanisms. So it's unlikely, in my mind, to think about this as a single bullet that would work. Second, I think the underlying conditions that we're dealing with here have many, many etiologies. How they originate and how we respond to them and then how we treat them, I think is going to need a toolbox of tools. So I'm encouraged that James is willing to put the money in. I don't know if, Joan, if it's 2.6, but it's a big number and it's a number we can't afford to build a new medicine. And our idea was to simply say, golly, could we do this on the lower cost end and still have patent protection? And we do have patent protection into the 2030s on a composition of matter using the unique combination of those 3 components. And that is an approach, but I'd say come one, come all.
Joan, over to you.
Yeah, I think all valid approaches, and I agree with Joe, all approaches, they're not even conflicting, they're sort of synergistic. You can approach this like James by targeting a pathway, starting with a rare disease that you know is dependent on that pathway, and expanding it to aging-related disease, or repurposing like Joe. But I think the key that it would be interesting to talk about with the other panelists is, once you get beyond a rare disease, if you want to do that as your pathway to approval, how do we grapple with these aging-related diseases that aren't confined to a rare population? It's confined to massive populations and are massively unmet needs.
Yeah.
And I think, you know, I lots of times will be listen to these panels and they go, oh, we just need to have really good clinical trials with the right endpoints. Those right endpoints, it's humbling how difficult it is to get the endpoints right when you're dealing with complicated, huge diseases that are affecting so many people. And I think we have so much to learn in the field about how to define them, which targets of aging biology do what to what population of aging people. It's massive amounts to be learned, and it's very exciting because there is so much that we still need to learn.
So I have a thought building on that, Joan, because I think that, you know, kind of pivoting away from the strategic way that we're each, you know, involved in businesses approaching the field, you're asking the key question, which I think of as the fulcrum point for the whole field, right? How does this field transition from being just another set of drugs that look like standard biotech companies to something really different? And the answer to this for me, obviously I have lots of thoughts and we could spend all day talking about it, but the answer to me involves scale. Because if we're going to take a shot on goal like the one you've described, targeting large populations with clinical trials and endpoints that, you know, maybe it's the right one, maybe it's not, we're going to have to try a few different things. My take on this is, if you're already starting with a multi-billion-dollar franchise on a new drug, that you have proof of mechanism of action, you can take a few more of those risks and start, you know, 2 or 3 phase 3 or phase 2b trials in parallel to test out those different endpoints. So I'm inspired a little bit when I think of the model that the checkpoint inhibitors that go after PD-1 followed in the oncology space, starting about 5 or 6 years ago with the first approvals, where, you know, let's take Merck's Keytruda, for example. Pembrolizumab was approved in, I believe, melanoma first, and it became rapidly what was looking like is going to be a multi-billion-dollar franchise in melanoma. And based on that, they were able to invest more and more money, and eventually had 26 phase 2 and phase 3 trials running in parallel for different indications. And they were able to justify that because they knew that some of the mechanism underneath it, the biology underneath it, was so strong. Some of these things were going to be successes and some of them were going to be failures. And by creating these huge data sets, spending billions, truly billions on these clinical trials, eventually the FDA looked at the writing on the wall from all of this clinical data that was being revealed, threw up their hands and said, okay, let's approve pembrolizumab for all PD-1 positive cancers in the late stage. And that sort of dynamic is how I think we're going to get the first blockbuster geroscience drugs. We'll test something for an orphan disease or a you know, some initial indication, say, hey, look, this is going to be a $2 billion franchise or something like that, some good biotech company drug, but then partner with one of the big pharma companies to run a cardiovascular trial and a diabetes trial and an Alzheimer's trial, and look at all of these different pieces, well, maybe weaving in 1 or 2 or 3 different phase 3s targeting geroscience endpoints, and then see what sort of data sets we can collect, being very creative 5, 6 years from now until they throw up their hands and say, hey, look, it looks like this thing is actually slowing aging, reducing risk of multiple diseases. We have to rethink the way that we prescribe this drug. That gets a little bit foggy towards the end point, but I think that the steps that we need to do to get there, from my perspective, making NCEs, feel like the right direction.
So from my point, Zan, I was trying to think about, well, okay, what can we do right now? Because patients are waiting, to quote the phrase you've often used. And if they're waiting, what are some simple things that could take place? And the idea of, and believe me, I'm not arguing against what Joan and James are saying. I'm just simply moving and saying, well, there are two things that I've learned in this space. Number one, every time I talk to the payers about underwriting the cost of drug that's going to address aging, their eyes roll in the back of their head. And the next thing is like, okay, you show me definitive proof that's cost effective of increasing age when I know people live longer are going to consume more of my budgets, and I'll pay for your drug. And so I said, well, boy, I don't know how big a hill that is, but I'm glad James has got a lot of money to go after that. So our thought was, well, why don't we turn this thing around and say, look, if there's a way to take the learnings that we have from our triple combination of leucine, metformin, and a very low dose of sildenafil, we actually have now developed the same impact cocktail, if you will, by using naturally occurring agents. And by doing that, we're now thinking that there is a possibility to develop in that middle space between the cluttered noise area of supplements and the legend drug space, a recognized opportunity, and then ask consumers to pay for it. And to put the proposition out there that the benefits, while not yet measurable, seem to point to improving insulin resistance, weight, perhaps liver fat, certainly blood pressure and triglycerides, and the combination of those is an implied value without making a disease claim. That's sort of our current thinking. I don't know if that's a bridge too far, but getting consumers to pay for something, I'm inclined to think is a solution that's a short-term solution. And it steps away from requiring the FDA to confer it, but does put on us the burden to demonstrate things that are really useful and safe, and for which there is not only a hypothesis of an MOA, but actually some demonstrated clinical evidence.
Well, we've got a lot of great questions that are coming in to your very point, Joe, and questions for both James and Joan as well. Let's take a pause as we bring in none other than Dr. Peter Stein, who is the head of new drug review at FDA, and clearly a terrific person to have join this panel. He will help us to understand what FDA has been providing as advice to sponsors who are pursuing age-related indications. But let me just say that Peter Stein, a fellow endocrinologist who brought a very distinguished career as head of clinical development in two major pharmaceutical companies before he came to FDA, now does head up the very important responsibility for the United States, the approval of new therapies. And so, Peter, welcome. We are delighted to have you. And I would just like to say, first of all, because you are very well known in the pharmaceutical world, but not necessarily by many other people on our call or our conference, could you maybe talk a little bit about the evaluation and approval of new drugs at FDA and your role in it?
Sure, and I'd be more than happy to, Zan. So, as you said, I'm the director of the Office of New Drugs. The Office of New Drugs sits in CDER. Just to step back a second, I think most folks know that the FDA carries on a number of different regulatory functions for foods, obviously for biologics, such as for tissue and gene therapies and blood products, and for veterinary medicine. So a whole range of different, and each of those are carried on within different centers. So I sit within the Center for Drug Evaluation Research, or CDER, and the Office of New Drugs is the office that really manages drugs from the early development, from early IND development through to NDA and BLA review and approval, and into the post-market space for post-market safety surveillance, which we work on with our colleagues in another office within CDER called the Office of Surveillance and Epidemiology. The process of reviewing new drugs is really one of applying our regulatory and statutory framework to the applications we see. And it basically is asking the question, is there substantial evidence of efficacy, of effectiveness? And is the benefit risk appropriate, which is how we look at the issue of safety. Does the benefit outweigh the risk? We consider that to be an appropriately safe drug. And if substantial evidence is provided, we consider that to be a drug established to demonstrate efficacy. And by substantial evidence, what we look at is, again, what is in our regulations and in our statutes, which talks about 2 adequate and well-controlled trials, or an adequate, well-controlled trial and supporting evidence, which is referred to as confirmatory evidence. Those are the standards that we apply in deciding whether a drug meets those regulatory standards and can be approved. Just to give you a high level, but it sounds like we might get into some of the specific details as the conversation goes on.
Oh, we will indeed. But let's talk about disease prevention and the misconception that FDA is not able to approve preventions for disease. Of course, FDA has a long tradition of approving disease prevention indications. And perhaps you could refresh us on that and the evidence that has been required for prevention claims.
Sure. Of course, the prevention claims is a broad range of different indications. You know, in a way, it's really part of the continuum of what we look at for the range of claims that different drugs have. There are, of course, many drugs that we approve for prevention, drugs for hypertension, drugs for lipid disorders, lowering LDL cholesterol, drugs to prevent transmission of HIV, many other such claims. You know, I think the framework is no different, which is really the question of what is the evidence for effectiveness and what is the evidence for safety. In a prevention claim, we have to think very carefully about the safety issues, because the question really is, how many patients will we need to treat to prevent the outcome event that the drug is intended to prevent. In a primary prevention setting, of course, there'll be many patients who may not ultimately get the event. That may be different in a secondary intervention type of approach, where the population is in a sense enriched for patients who are more likely to go on and get the event. So our tolerance for safety, for example, may be different in a primary prevention, otherwise healthy population compared to a secondary prevention population that are more likely to both see the benefit and to reduce the risk of that event occurring. So really the questions are the same when we are looking at a prevention indication. What is the evidence for benefit, and how does that relate to harm? One simple way when you're looking at prevention is to look at statistics like the number needed to treat and the number needed to harm, and what those benefits are. How significant is the prevention of that particular event that the drug is intended to prevent relative to the harm the drug might do? What is the safety issue, and how do we weigh that? These can be very thorny issues. For example, drugs intended to prevent cardiovascular events, some of the anticoagulant drugs or antiplatelet drugs, have bleeding risks. And so we have to think very carefully about the ability to prevent the events such as a myocardial infarction or a stroke, but also the risk that a patient might have a bleed. And how do you weigh those things? That can get to be a thorny balance. It's not just a numerical question, it's also the impact on people's lives of the events that are prevented, as well as the risk and the impact on people of the harms that might occur with that drug. So that's where the prevention issue becomes challenging. You know, what are we preventing, and how much benefit does that offer a patient, and what are the risks the drug has, and how impactful might those risks be?
Now that you mention cardiovascular prevention claims, of course, those have been based on composite endpoints, composite of mechanistically related clinical endpoints. And that is done to facilitate the ability to detect a signal sooner than would be possible if you, for example, insisted on a single endpoint as the primary outcome. As you know, there is a proposition that we use a composite endpoint for approving age-related indications, so-called health span indication. Could you talk about making that jump from the well-established composite endpoint for cardiovascular prevention or any other claim related to cardiovascular outcomes, and what we're attempting to talk about today, that is endpoints that could get a age-related indication across the line?
Sure, that's quite a bridge to go over. So of course we use composite endpoints in a variety of different settings. You might consider them in sort of two groupings. One are composite endpoints where different patients in a study might get a different endpoints, and we count them together into that composite endpoint. And then, of course, many composites that are focused on the effects of the drug on an individual. Those that are, for example, clinical outcome assessments, things like PANSS scores for schizophrenia, or HAM-D for depression, or the ACR20 for rheumatoid arthritis. These are also composites, but they're assessed within individuals. But we are really asking about the composites that are assessed across a study population. And of course, the idea is in a sense twofold, and I think both of these potentially are applicable to the potential for an age-related composite. One, as you mentioned, is to enhance study power. If we know that a drug has potential effects on a number of different endpoints, and each of them relatively infrequent, of course, by combining them into a composite, we will have more events, which would allow for a smaller and potentially shorter trial to occur. But not infrequently, we also have composites of a similar sort where we're not quite sure which of the endpoints might be most effective. A new cardiovascular drug, for example, might have a greater impact on reducing stroke, or a greater impact on reducing myocardial infarction, or might not change either, but still might reduce cardiovascular death or congestive heart failure, for example, as some of the more recent classes of drugs for diabetes have been able to demonstrate. And so when we don't quite know which of those endpoints will be benefited, we can also create a composite that may capture the benefit without us having to know in advance which of those will be most effective. But one key principle that we have to think about with that is that the components of the composite need also to be relatively similar in their impact. We wouldn't want to combine an endpoint, for example, that had a relatively limited clinical impact with an endpoint such as cardiovascular death, which would, of course, have such an impact. And so we have to also think about the components of the composite. And even when we analyze the composite, we also have to understand what drove it. If the trial is successful and the composite demonstrates a meaningful and statistically significant reduction, what were the components of that composite and how were they individually affected? We've certainly seen examples where, for example, for MACE, the major adverse cardiovascular events, where there may be a particular effect on myocardial infarction, but no effect or even a slightly numerically unfavorable effect on stroke and cardiovascular death. And so we do need to think about that. When we think about age-related composite, the same principles apply, which is we have to think about what the events are and how clinically meaningful they are. When we think about approving drugs, we think about endpoints that are clinically relevant, clinically meaningful. Why would a patient take a drug if it didn't affect how they felt, how they function, or how they survived? And those are the endpoints that we look for. So when we think about an age-related composite, you have to think about what are the components of that that are clinically relevant, that are clinically meaningful to patients? What are we trying to prevent? You know, I think as you design these kind of composites, it's going to be really important to be informed by the earlier work on that particular drug. You know, the problem with putting together a composite is that if only one of the components is positively impacted, the power of that study might actually be deteriorated by using the composite as opposed to enhanced. And so you have to think very carefully about what are the components. They should all be relatively similarly clinically relevant. We wouldn't want to combine a minor benefit with a major benefit. And we also want to make sure that they are likely to be impacted by the drug. At least a number of them would be likely impacted by the drug as opposed to just one of them benefited. I think that there is the potential to come up with clinically meaningful changes that a drug might have that might be age-related. But another approach to this, and perhaps it is in the form of a composite, is to imagine that a drug that targeted age-related progressive deterioration will lead to patients having improved function. And so an endpoint such as preventing patients from requiring higher levels of care, or maintaining independence longer, or preventing hospitalizations, which might be sort of integrated across the various components that the drug affected, such as cognition or motor function or prevention of comorbidities, all might roll up to keep a patient functioning independently longer, which certainly would be a clinically meaningful benefit to patients. And that also could be put together as a composite, such as prevention of higher level of care, longer maintenance of independent activities of daily living, prevention of hospitalization. That might be a composite that could be considered as well. So there's lots of ways of approaching this, but the one point I would make is that the composite has to be very well thought out. The components have to be clinically relevant and generally similarly clinically relevant. And you'd want a drug that would affect multiple of the elements of the composite and not just one, as that would probably not be an optimally designed trial. Remember that you design your trial to be enriched in patients who are likely to either benefit from the drug or have the events the drug is intended to prevent. If that's what you're looking for, you have to design a composite that allows you to pick the right population so the trial can be not overly long and not overly large.
Peter, that's very helpful. I'd like to bring my co-panelist in to comment or ask you a question. Of course, we have only a few minutes, but I hope that at the end, you might say a few words about how do we continue the conversation? How do we ultimately get to a guidance for sponsors who are seeking some kind of health-related or health span claim? So while you're pondering that, let me ask Joe Cook, would you like to come in here? Or Joe, I see you're on mute. Joan, why don't you, well, there you are, Joe. I caught you with a mouthful, I see.
I was just finishing a cheese and cracker.
All right.
A low-fat version thereof.
This is the opportunity of a lifetime
Yeah.
to speak to the head of new drug development or new drug review.
Peter, first of all, thank you for your leadership and what you've been doing. We've struggled with our approach because we saw a benefit across multiple factors. And in the aggregate, it seemed to address what has historically been called the metabolic syndrome or syndrome X or whatever, all of which, Zan and I have talked a lot about this. I'm not sure that addressing that per se would satisfy the need for something that was protective for the long term, but it seemed to us to have utility. If you could lower blood pressure and improve insulin sensitivity and improve weight in a composite manner, all of which individually may not be stellar or spectacular, but taking as a composite of those metrics, we do believe that people will be able to function better and live longer. But the proof of that is pretty difficult to tease out. So I don't know if you've given some thought to the kind of measures that are generally associated with metabolic syndrome, or what used to be called that, if such a composite might be a step one toward a geroprotective agent, knowing that if we improve those underlying factors, we're most likely going to improve overall health in some way.
So now I'm going to sound more like a regulator than a drug discovery person. I think that what is needed would be sort of a strong hypothesis that went into the design of a strong outcome trial. I mean, I think the hypothesis is certainly reasonable if we affect in a smaller way multiple factors that are all thought to be contributory towards outcome of cardiovascular events, for example. There may be other outcomes that are also potentially thought to result from the range of abnormalities that might constitute metabolic syndrome. And I think if the hypothesis is that modest changes across a range of factors will cumulatively provide a benefit on cardiovascular outcome, for example, I think you would still need to demonstrate that. I think you need to show that the cumulative effect was one of a clinically meaningful reduction in cardiovascular events that were relevant cardiovascular events. Now, that does imply a large trial. That does imply, trials that are the typical 6 to 10 or 12,000 patient trials. Obviously, you'd have to design it accordingly. But I don't think you can. I don't think there's an immediate intermediate step where simply showing small benefits across a multitude of endpoints would be sufficient for registering a drug for prevention. Now, again, we do have surrogates that we accept. LDL cholesterol, for example. A reduction in LDL cholesterol is a surrogate, a validated surrogate that we allow for to support full approval of drugs for hyperlipidemia, for hypercholesterolemia. If the drug had a sufficient effect on blood pressure, for example, that also could be a route to registration, even if it had other positive effects that might ultimately lead to a better cardiovascular outcome result, but you can register the drug in an intermediate way using blood pressure or LDL cholesterol. However, small changes in other biomarkers would not in and of themselves be sufficient to register a drug.
No, I accept that. I should have stated that in one of our studies, we did show a greater than 5 millimeters of mercury of effect on blood pressure, which I believe is the current minimally accepted standard. But when you think about marketing another, quote, antihypertensive agent, good luck on getting anybody to really pay anything approaching what it would cost to even get it submitted.
I mean, I understand the quandary. As I said, there isn't, there isn't an easy path to getting a broader indication, but certainly a drug which starts off as a drug that improves at parameters such as blood pressure in a sufficiently meaningful way could be registered as such, and then an outcome trial, once demonstrating broad benefit, would be able to be registered to describe that benefit if the trial was designed appropriately. You can make the argument that that's really the course that occurred with SGLT2 inhibitors, which appear to have a benefit on a number of different outcomes of relevance, both renal and cardiovascular, but were registered, of course, as drugs to lower glucose, to improve, to treat diabetes. One could argue that of course, is a surrogate HbA1c, and was an intermediate, if you want to look at it that way, indication, ultimately demonstrating cardiovascular improvement and demonstrating reduction in congestive heart failure hospitalizations, and then demonstrating reduction in renal progression, first in diabetics and more recently in diabetics and non-diabetics. And so you can see that as a sort of staged approach to demonstrating benefit for a drug. But the claims that you can get would, of course, reflect the studies that are done.
Helpful. Joan, you've spoken in favorable terms about the interactions you've had with FDA officials who have been helping you to pursue your indication that relates to improving immunofunction and actual clinical outcomes related to that. Perhaps you could give our audience a sense of just what the process is, and maybe interact with Peter in how you came about developing what was essentially a de novo indication, one that hasn't been approved before.
Sure. So to step back, there had been data that inhibiting a protein kinase called mTOR has beneficial effects on the function of multiple aging organ systems and increase health span and lifespan. So we started by just seeing if we could improve immune function in older adults by giving them low doses of mTOR inhibitors. And again, to Peter's point, we tried to use doses where the safety risk would be low and would be outweighed by the benefits. And what we found was that these low doses of mTOR inhibitors enhanced antiviral immunity. So antiviral gene expression, interferon-induced antiviral gene expression was increased, and there was a decrease in the incidence of multiple viral respiratory tract infections in a phase 2b trial. It was interesting because in the phase 2b trial, the FDA wanted an endpoint of lab-confirmed respiratory tract infections. And we hit that, but then on the phase 3, they changed their mind and said, in the real world, nobody has, and this was pre-COVID, has the cause of their respiratory tract infections laboratory confirmed. They just know they have an infection, and usually it's viral and there's nothing you can do for it. So what you have to have an endpoint for phase 3 is relevant to how people feel and function. And so it has to be just respiratory symptoms, irrespective of whether they're laboratory confirmed. It made sense what they were saying, but it turns out older people have a lot of non-specific respiratory symptoms. So it became a very noisy endpoint where it's really tricky to tell what respiratory symptoms were due to an infection versus due to something else. And people who knew they weren't sick were getting triggered for the endpoint, and we didn't hit the endpoint. So I think we have to, this is what I'm saying, there's a learning curve to go in an older adult population that how they feel and function has to be a little bit fine-tuned. And there was also a huge placebo effect. So people who were getting placebo were saying they had never had fewer respiratory tract infections. Their respiratory tract infections were milder than they ever had been before. I mean, it was remarkable that just taking placebo truly has a dramatic effect on how people feel and function. And we've seen this and all sorts of aging-related symptoms that people on placebo feel a lot better. So it's a complicated thing to get right, but the agency has been very good about saying, look, we've got to learn this together. These are new areas. And then I guess my last thing is I really think it's right to succeed as our whole field. We really have to start identifying who is at risk of getting the endpoints that we're looking at and enriching for that population, and also who are the people who are really going to benefit. Because, for instance, people who already have good antiviral immunity don't need a drug that boosts it, whereas the people who have deficient antiviral immunity, like we're seeing with COVID, the people with deficient interferon responses, they're going to get into trouble with COVID. So there's a complexity and a learning curve that I think the field needs to start tackling so that we can get, a good positive trials.
That's a really interesting, I think it's really also a very informative experience. And you mentioned a couple of things which I'll just underline, one of which is the design of the trial, making sure that you are enriching the patient population as appropriate to patients who really could potentially benefit. As you pointed out, if you have appropriate markers, now one has to be careful with that. You have to be able to use tools that are clinically applicable in practice, because obviously if you use a test to enrich the population that's not available as a companion diagnostic or already available widely, that can be troubling in terms of trying to design a trial that will be relevant to clinical practice. But the other point I would make is that we really encourage the kind of interaction between the division and the investigators that it sounds like you had. The folks in our antiviral group and in our pulmonary and allergy and critical care group, I mean, we have some terrific subject matter experts. But again, they really look at their role as collaborating with the investigators to try to design the trial in the most sensible way most clinically appropriate way and in a way that will give you the most robust result. And I think that the opportunity to sort of push back on them and to have a really good and robust discussion is really important. If you feel like, we don't quite agree with that, I think it's very reasonable to push back and really make sure that you engage with them in a really collaborative discussion. At the end of the day, the agency is going to give its advice, and the investigator, and what we do give is at that stage is advice. It's not an absolute rule unless the trial design isn't safe. But again, the intent is for us to be collaborative and to have a back and forth, and not to absolutely just lay down the law, as it were, but to really lay down what we think is our opinion, and then to hear what the investigator thinks.
Very helpful. James, anything that you would ask of Peter? What do you need as a venture capitalist who's looking at investing in assets that ultimately are aimed at health span indications?
For me, I think the biggest question is around this topic that we've touched on a couple times around timing, and the trend that I see overall in this field of age-related diseases is moving closer and closer to primary prevention, which Peter talked about a little bit, especially in fields like Alzheimer's disease, where we've just failed over and over again to interrupt the pathology of the disease once it's already developed. Groups like Eli Lilly and Roche are going earlier in disease pathogenesis, and I think that the whole field that we're talking about today, this geroscience field, or metabesity, like you like to call it, Sam, is a reflection of this thinking applied to so many of the different diseases of aging. One of the reasons I stepped into it was because looking at the results in animal models, the efficacy data is so profound at the ability to prevent all of these different complications at one time by targeting this damage early, that there's something that clicked in my brain that said, this is the future of how we have to do medicine. The challenge here is that as we move further and further into primary prevention in a world that is operating on patent timelines to approved drugs, there is this huge challenge of how long the trials are actually going to be to see that endpoint, right? So I think Joan just described a trial that all of us in the field thought was really, really brilliant, because you could measure this endpoint very quickly. And this mTOR effect was one that seemed to boost the very flexible immune system quite rapidly. Over the course of a few weeks or months, you could already see an improvement. But I think many drugs in this space are going to be much slower acting than the one that Joan was working on, where you might have to treat 5 years or even more before a person would be diagnosed with Alzheimer's disease or mild cognitive impairment in order to see an effect, right? So my question for Peter is, how does the agency think about this shift in medicine? And is there a role for maybe doing more to work with industry partners to increase the number of phase 4 trials, where we could, and I know this has been talked about a little bit in the Alzheimer's space, where you could do approvals from large phase 3s based on initial signals, some biomarker effects, but have the group that does this, that does that trial, commit to long phase 4 follow-ups, not just measuring increased safety, but also looking for those hard endpoints like, admissions to the hospital, diagnoses of different diseases, et cetera. I think a direction like that, not in 1 year, but through collaborations over the next 5 years, is really what can move this field forward. And I wanted to get your reaction on that thought.
Well, I mean, there is an avenue for a structure of that sort, which is accelerated approval. And what you're describing is really accelerated approval. So I mentioned before, that we do approve drugs based upon validated surrogates for full approval: blood pressure, HIV, RNA, Hep C RNA, LDL cholesterol, A1C. Those are endpoints that we use for full approval, surrogate endpoints we use for full approval. An accelerated approval can be based upon an endpoint which we consider to be reasonably likely. or in an intermediate clinical endpoint. I won't get into that further, but a surrogate that we consider to be reasonably likely and then has to be confirmed in a large clinical trial establishing clinical benefit subsequently. The issue is that reasonably likely doesn't mean hypothetically possible. It means, that there's a good body of evidence that particular endpoint reflects and will predict clinical benefit. That's really, I think, the challenge, which is what would that endpoint look like? Certainly, we are open to those discussions. We have a whole group that works on biomarkers and qualification of biomarkers. And so we'd be open to discussions about what such an endpoint might look like. But again, the standard for reasonably likely is not a sort of just a body of limited information or epidemiologic information. It really is an endpoint that we have confidence in. It doesn't have to be the kind of body that led to LDL cholesterol, for example, being accepted, which was a huge body of information, both preclinical, animal model, epidemiologic, and many trials that demonstrated the relationship between lowering LDL cholesterol and improved cardiovascular outcome. But there needs to be a fairly solid body of evidence, and I think that's really so if you're asking, is that possible? Yes, I think that is possible. But the problem is that I think where endpoints are not well established as likely to predict benefit, but more sort of have a hypothetical relationship, and maybe there's some epidemiologic or small studies that might not convince us. I also would add one other point here, which is that we also worry about the size of the population that will be treated. Alzheimer's obviously is a disease where you're talking about millions of patients who may be worried and want to take such a medication. So it exposes a huge population to a drug that hasn't been established to have benefit. And so much of our accelerated approval is in oncology, for example. We have many drugs that are approved based upon either response or progression-free survival. And also in rare diseases where, again, it takes many years to see the interruption of the progression of the disease, but there are very logical biomarkers that reflect the genetic defect. More difficult, I think, in something like a disease where that's not the case. On the other hand, there is a new Alzheimer's guidance, which I refer you to. And in prevention, we recognize that because there isn't yet clinical dysfunction, patients may still be functioning reasonably well. There is the ability to use cognitive testing, particularly multiple different parameters of cognitive testing, neurocognitive testing, that is in that guidance. And so I think there is some flexibility in how one can get a prevention indication for cognitive decline in Alzheimer's. But as I said, I think accelerated approval, which is what you're framing, is a challenge because the endpoint still has to be reasonably robust. It's not 100%, but we have to have confidence in it.
Really great answer. Thank you. Yeah, I think for me that kind of sums out to, it means that there is this priority in the space in this field to start having that conversation amongst ourselves with the FDA almost now to think about how we can generate the appropriate body of data to start having these conversations, because I think that there will be continued opportunities like the one Joan told you about, like the one Joe told you about, where you can impact one of the registrable biomarkers today, or you can design a shorter iteration trial, but sooner or later there will have to be sufficient data package so that I could sit down with you and your team and say, this is what we would like to do. This is, we'd like to sign up for this 7-year trial, but our goal is to talk about an accelerated approval at 3 years or 4 years or something like that, where this can become real for the wider industry.
I mean, the only other quick comment I would make, just briefly, is that I think these are the kinds of things, and I know there's already lots of collaborative consortial efforts across this space. I can't underline how important that is, because I think in other spaces where there's been a lack of adequate biomarkers, the field can get very fragmented with different companies and different academics and different even government groups. working not really across purposes, but not in a unified way. And so consortium, public-private partnerships, working up, in the pre-competitive space, I think is so important here to prioritize what those endpoints might look like and to work collaboratively across groups to develop them, because making progress on these kinds of endpoints requires a very comprehensive and cohesive effort. And so that's the one thing I really would encourage is, if everyone does a go it on your own kind of approach to this, it's going to take many, many years before you're going to find predictive prognostic biomarkers or even accelerated approval of reasonably likely surrogates.
Zan, I just, Peter, I'm so glad you said that, because I really do think that one of the outsized benefits of the Metabesity conference is to curate and cultivate such collaborative discussions across the whole world. I mean, if you look at the panelists who are on here, they represent a wide range of things. We're not going to agree on a lot of things, but if we could recognize the burden of what do we need to think about in the context of what the FDA is trying to avoid, which is approving something that has an unknown risk for a benefit that's very far out in the future, how can we contribute to defining a better characterization of that risk benefit, even if it's an interim or a transitory nature that we're heading toward these, your moonshot, James, when you find it. I hope we contribute to improving the life expectancy of the people who are sitting waiting today, either with Joan's work or some of our work. So Zan, just say, let's keep this conference thing going, but I think you have a curation duty here that's really special and unique.
Oh, thank you for that. It's a perfect segue into the last question for Peter. And Peter, you've been so generous with your time this afternoon. This is a federal holiday, and we really appreciate your making yourself available.
Well, with COVID going on, I can tell you that none of my very few of my organization are taking the full day off. So
yeah, well, we know
lots of people working hard.
And just to say thank you for your service, because we know that many of the divisions are really working 24/7, particularly to respond to the COVID pandemic. So thank you for that. Can you just leave us with any kind of suggestion or guidance on when a guidance could be available?
Well,
or the process. That's not fair. Maybe just to describe in a few words what could be the process that would get us.
Sure. Well, the one point I make is that there are, I think, some guidances that are relevant to this space and I think are already informative. You were asking before about composite endpoints. There is a very good guidance on multiple endpoints, which includes sections on composite endpoints. And I think that's I think provides some information that's very relevant to this. We just put out a guidance last year or updated a guidance last year on adaptive trial designs. And that I didn't say much about that, but that's another approach here, particularly when we're not quite sure about what the patient population or the endpoint might be. One can do an adaptive phase 2-3 design that might mitigate some of those risks by using ongoing information from the trial in adapting the design of the subsequent stages of the trial with appropriate statistical control. So I think that's another guidance that's relevant to it. We have a guidance on biomarker qualification that we put out relatively recently. I'm sort of skirting your question to a certain extent because you're asking more about age-related things. What I'd say there is that, as our guidances are typically when we've had experience with working with programs, and typically more at the program level, have sort of some clarity as to what our thinking is and what we're going to require, what the science externally tells us, and those come together towards a guidance. I think at this point it might not quite be ripe for a guidance on this. But as we have more interactions, such as the one we talked about with the mTOR inhibitor, it informs our thinking about what's required, and I think it allows us to have more relevance to put into a guidance. We also have meetings that are purely intended to be interactive scientific meetings that are called CPIM meetings, and those are another form in which we're often informed and we get a sort of a scientific interaction. It's really these kind of multiple different interactions that sort of inform us, give us the background, and help us think about what's required. But as we do that, that's really how we begin to generate a guidance. And you'll see that also sometimes we will put out a draft guidance early on. Draft guidances really are meant to seek input sometimes. So we'll actually, in draft guidances, you'll sometimes say, FDA's thinking on this is evolving, comments, put comments to your thinking in the docket. So sometimes we actually ask for input through the vehicle of a guidance. So I can't say I would see one in the very near future, but as we more interact with folks interested in this area, I think that will stimulate our thinking, and likely that's the pathway to a guidance.
Fair enough. Dr. Peter Stein, thank you very much. We really so much benefit from your participation, and we look forward to the next time. I thank also my colleagues, fellow panelists.